So thanks for being here this morning, everybody. I have some bad news. We're not going to be talking about pediatric research this morning. Maybe for some of you that's good news. We have planned on talking about the pediatric research integration and what that means for our work together in the coming years. And we're just not to the point where we can do a whole grand rounds on that topic. There's lots and lots and lots of work underway. And hopefully soon we'll be able to share more about that and give you all a lot more details. So as a backup topic, but I hope something that will be really compelling for folks, I thought I would talk about using race in clinical care and giving sort of a genetics background to that question. So for those of you who are on the research conference recently, you may be thinking, Kyle, I've already heard you talk about this. But I did want to say that the earlier topic that I did for research conference was really focused on our research practice. And I spent a lot of time talking about systemic racism, and Jennifer Porter, who presented with me that day, talked about systemic racism. I'm not going to talk a ton about research, and I'm not going to talk a ton about systemic racism. Instead, I'm going to take a slightly different perspective on this topic today, and instead talk about genetic ancestry and really dig down so that maybe we can all understand what genetic ancestry means, that the test that you get from Ancestry.com, what that means, and then hopefully disentangle a little bit about that kind of information and what it means to see patients and try to think about what race means in the clinical context, okay? So general overview for this morning, I'm going to start out by talking about genetic ancestry. Then I'm going to talk about the question, should we use race as a proxy for genetic ancestry in our clinical practice? And finally, recommendations for clinical practice. What can we do differently in light of this information that we're going to be exploring today? Okay, so first, genetic ancestry. I have a video. I will say I have been a faculty member at the University of Louisville since 2012. And this is the first time in that entire period of time that I've ever put a video into a PowerPoint, because they're so unpredictable. And as you all know, you've probably seen dozens of people have them fail. So anyway, with that preview, I'm sure this is going to go great.
Growing up, we were German. We danced in a German dance group. I wore lederhosen. When I first got on Ancestry, I was really surprised that I wasn't finding all of these Germans in my tree. I decided to have my DNA tested through Ancestry DNA. The big surprise was we're not German at all. 52% of my DNA comes from Scotland and Ireland. So I traded in my lederhosen for a kilt.
Ancestry has many paths to discovering your story. Get started for free at Ancestry.com.
Okay, I show that video not just because this guy's name is Kyle, which I'm offended by because this Kyle doesn't understand Ancestry, and also because I just want to point out how this video, in kind of an insidious way, is really trying to convince you that your genetic ancestry defines who you are, or defines your identity, your understanding of your heritage. Of course, they're doing that because they want you to buy their product. And to be honest, their product is an excellent product. And I can talk a little bit more about that during the course of this presentation. The problem is the marketing and the way that the information is framed causes us, it reinforces assumptions that we already have about race and genetics. And so the next probably 20 minutes, 30 minutes of this talk is going to be focused on trying to convince you not to think of race as a genetic, as having a genetic or biological basis, okay? For some of you, this might be a little bit in the weeds, but I hope that it really helps bring to the surface the truth about genetics and race, and hopefully can help us think about our clinical practice in a slightly different way.
Growing up.
Okay. So what can genetic ancestry tell us about our identity and our heritage? So in order to broach this topic, I first need to say just a few words about our genomes, because many of us learned about genetics from a Mendelian framework. So we learned about alleles, we learned about mutations, we learned about how mutations cause diseases. However, there's actually like 99.999% of the genome that medical school never touches on. And so I really wanted to give you some really brief pointers about what the genome is like as a whole and not just from a clinical perspective. And then that will help set up as we talk about how ancestry tests actually work. Okay, first thing to remember is that important parts of our genomes, they don't vary much. You know, through the process of evolution, the important parts that really determine, you know, how our bodies are made, how our body functions, how our body is adapted to the environment of, you know, this planet that we live in. We actually, things that are important tend to not be that different among us. So in fact, we share some important parts of our genomes with plants, fungi, insects, etc. You can go back, you can find very important parts of certain proteins and enzymes in our body that we literally share with bacteria. And that, I mean, that shouldn't be that surprising, right? Because if something was so super important, if it changed, that would not be compatible with life, right? So the variants that cause diseases, and we don't use the word mutations anymore because it implies that there's something inhuman or wrong about them. And in fact, these are many of these changes are the kinds of things that, depending on the environment we live in, might actually be adaptive. So we just refer to those as pathogenic variants or disease-causing variants. So these are typically those that occur de novo, right? So our children that we take care of who have autism as a result of a genetic condition or epilepsy or you know, intellectual disability, all of those kinds of things that we know have a genetic basis, those are disproportionately caused by de novo variants, variants that occurred during the process of their formation as an embryo. And that again should not be that surprising. If those changes in the genome were so harmful, it'd be unlikely that they would be passed down from parent to child, right? We would see those same symptoms in the parents. Diseases that are genetic can also be caused by rare variants that are inherited by a small number of people. So I know much of what we learn in pediatrics about genetics has to do with these rare variants that are in populations. A small proportion of the population carry these. In many cases, a very, very small proportion carry them. And when, by random chance, a biological mother and a biological father have a child, and there's the child inherits the same variant from both parents, or a variant in the same gene from both parents, that can lead to a recessive condition, right? So in general, those rare variants are only in a very small number of people. They're not very good for determining a person's ancestry because they're rare, right? They don't reflect an entire population of people. And then finally, I just want to say, kind of repeating here, truly harmful variants aren't common, okay? So I put a parentheses, or I put an asterisk there just to bring up what you're thinking about probably, which is sickle cell and cystic fibrosis, right? Those are two truly harmful variants that are common. Actually, they are the most common disease-causing variants in the human population. And the reason that this statement is still true despite those is that I said truly harmful variants. So as you know, there's an evolutionary explanation for why both cystic fibrosis and sickle cell are relatively common. They can be protective for infectious diseases. In the case of cystic fibrosis, it's thought to be typhoid, and in sickle cell, it's thought to be malaria. So evolutionarily, being a heterozygous for what we call a disease-causing variant is actually protective from those infectious diseases. So in that case, there is a biological drive for why those variants are relatively common. Truly harmful variants that are harmful no matter what are not common. Okay, let's talk about common variants, because most of this talk is actually about common variants. So common variants are changes in the genome, or differences in the genome is probably a better way to think about it, that are present in, typically the cutoff would be something like 5% of the population. So common variants typically only have a very small effect on health, and in fact, they are often biologically silent. We can go into all sorts of reasons that changes in or differences in genomes might be biologically silent, but just remember we have about 20,000 genes. That is nowhere near covering all of the genome. There's tons of garbage in the genome that has no biological effect. And as you all know, as many of you know, there are changes that occur even within genes that don't end up resulting in a change in a protein. So common variants are often have literally no biological meaning. They don't show up in a human being at all. And these are the kinds of genetic differences that make all of us distinct from one another. They're the kinds of differences that if you were, being investigated for a crime and they were going to use genetic information, these kinds of common but also silent variants are typically the things that are used in those kinds of identity tests. Common variants are typically present in every population throughout the world, but they occur at different frequencies. And the term for this is allele frequencies. So this is extremely common, or this is extremely important, and this is actually what I'm going to talk about for the rest of this piece on genetic ancestry. Common variants are present in every population throughout the world, but they occur at different frequencies. Okay. So in order to talk about allele frequencies, we first need to talk about a SNP. A SNP is not something that we learned about in medical school. That's not the kinds of thing that we talk about. But a SNP stands for single nucleotide polymorphism. So those words probably, if you understand what a polymorphism is, you probably understand now what a single nucleotide polymorphism is just from the words. A single nucleotide polymorphism, or a SNP, is a single nucleotide in a genome, in this case a human genome, which can be different. And there's kind of a weird dynamic that I don't understand. There's probably human geneticists who do understand this. But the vast majority of these are biallelic, which is to say you only have A or G. Even though there's also T and C, they don't exist at SNPs. So the vast majority of these single nucleotide polymorphisms are biallelic. There's only two options. Some are triallelic, less than probably a third, maybe more like 10%. So we have many of these SNPs across our genomes, over 500,000 at least. And these are sites where we differ from one another. When you hear people talk about 99.9% of the human genome is shared by everybody, what they mean is there's SNPs and then there's everything else. So SNPs is where people differ. Okay. When you think about a biallelic or triallelic SNP, it's important to remember that typically both or all three of the variants found at a SNP location are typically present in every population throughout the world, but they occur at different frequencies. Okay, so just restating what I said earlier, but this time using the SNP terminology. Okay, there's next slide will help make this point. So you can see here's 4 populations. This is just a single SNP that we're talking about here. Some people have an A at this single location. Some people have a B. And in the different populations, the percentages are different. Population 1, half of people have A, half of people have B. Population 2, 25% of people have A, 75% of people have B in that population. And then we have an example of two populations where only A exists in population 3 and only B exists in population 4. Okay? So these are the allele frequencies. You can say the allele frequency in population 1 for A is 50%, right? Okay, these differences are the basis for genetic ancestry estimates, okay? So this is how, there's two methods that companies like Ancestry.com or 23andMe use for giving you an estimate of where your ancestors came from. And method 1 is based exclusively on the proportion of variants at multiple locations across the genome. So imagine just for a second that I was tested for this particular SNP that's shown on the slide. And at that location, the test reveals that I inherited an A from my father and a B from my mother. So which population am I from? Just think about that for a second. Am I from population 3? Clearly not, right? I inherited a B from my mother. Am I from population 4? No, I inherited an A from my father. So if I was going to try to guess which population I was from, I would say population 1 or 2. And actually, chances are a little bit better I'm from population 1. Because A and B both occur at equal frequency, okay? And if you think about it, you can actually take the probability that I'm from each of these 4 populations, you can assign a number to it. What are the chances I'm from population 3? 0%. What is the probability I'm from population 4? 0%. And then, I don't know the real number, but population 1 is a pretty high probability. Population 2 is slightly lower, okay? Repeat that 100,000 times, okay? So at Ancestry.com and 23andMe, those kinds of companies, they look at over 100,000 SNPs, and they make this kind of estimate 100,000 times. And then essentially what they do is they map out what combination of ancestral populations most or best match these probabilities, okay? That's all it is. So I'm going to drive this home multiple times, and I apologize for being repetitive, but the key thing to notice here is that even though Ancestry.com and 23andMe are able to give me a relatively meaningful estimate of where my ancestors' populations are from, they are not using anything that is substantively unique about any of these populations, right? There's no UK or United Kingdom genome, right? There's no gene that's only found in populations in Germany, right? We're not unique in that way. Populations are not unique in that way. As a matter of fact, the fact that these allele variants just vary from population to population, and in general, most populations have both, it tells us that, in fact, the human gene pool is a spectrum, right? There's no hard and fast divisions within the human population. We're simply a spectrum. And so even though we look at that report and say, well, 52% of my genome seems to come from populations that today live in the United Kingdom, Ireland, and Scotland, that doesn't mean that there's anything different about the populations of the UK, Ireland, and Scotland that's completely unique from any other population, right? And importantly, the kinds of things that they are using to determine my ancestry or estimate my ancestry have nothing to do with disease, right? These are, by definition, common variants. If they were rare variants, they wouldn't be very helpful because those are rare. They have to use common things. So they're telling me, they're estimating my ancestry with never using a disease-causing variant, right? They're not paying any attention to the disease-causing parts of our genomes or the parts that, you know, keep us healthier or whatever. Okay. Method 2 is based on what's called a haplotype, okay? A haplotype is a pattern of sequential SNPs found on a single copy of a chromosome. So either the copy that came from a person's mother or the copy that came from their father. And this is essentially very similar to method 1, except for it's using patterns that occur relatively uniquely or within populations. So haplotypes occur with different frequencies in different populations. And in some cases, you know, where allele frequencies are low, you might have a combination that's found in only one population. So that can be somewhat identifying. Again, based on the common differences in our genomes, but just looking at the patterns of those common differences. So this one's a little bit hard to conceptualize, so I came up with an analogy which I hope will be helpful. Okay, so this name, Kapule, is a Hawaiian name. And as far as I know, the name Kapule does not occur in any other population around the world, okay? So if you met someone who's named Kapule, you can guess that either they're from Hawaii or their parents had some kind of affinity for Hawaii, right? Okay, the key thing to remember here is that the letters K, A, P, U, L, E in that order are what's distinctive, okay? So there are other names that have those letters, like Kaplan is a traditionally Jewish surname. Kapel is a German surname, and Jokul is a Scandinavian name. So if you look, you can notice that KAP in order, those are relatively common occurrences in names, and they overlap with this name Kapule, but they are for completely different populations, right? So the fact that KAPULE in as a pattern is distinctive to Hawaii doesn't mean that the letters, the individual pieces that make up that pattern, are uncommon. They're actually can be quite common. And you can see Jakul with this ULE at the end matches Kapule, completely different languages. Again, the fact that this E at the end of a 6-letter name, that's not that uncommon, right? There are many names that have that. So the whole point here is these are common letters put together in a relatively distinctive way. So another piece that is important to remember is that even though these are kind of sequential in the genome, they occur in this order, they're not adjacent to one another. So you can see here KAPULE is mixed up with lots of other T's and C's and G's and A's. And those other parts of the genome might be completely identical among every human, right? So we just have these SNPs scattered throughout a portion of a chromosome, and they occur in this order, but they're not next to each other. And the reason I wanted to point that out is it's not like you're making a major change in a whole section of a chromosome. These are just common differences that occur that are kind of spread out throughout a chromosome. So haplotypes are distinctive patterns of SNPs that are individually common. The individual SNPs are common throughout the entire human population, but they're typically not adjacent to one another, and they're typically not relevant to health or any other phenotype. Okay? So those are the two methods. When Ancestry.com first opened, they used method 1. They have switched to method 2 in that 10-year period. So if you send your sample off to them, that's how they're going to give you an estimate. Just want to be repetitive in order to drive home this message. Ancestry estimates that you get from these services are based on SNPs, alone or in combination, that are individually found in all groups. Okay? The reason I wanted to really drive that home is that the fact that companies can trace ancestry using genetics does not mean that these groups are particularly distinctive from one another. As the human species, we are actually quite similar to one another. And the differences that vary across populations are typically not relevant to health. Okay? So I hope you can see where I'm going here. I hope that was useful. So let's dig into one example because I think it really starts to drive home the clinical implications of these insights. We're going to talk about US patients who self-identify as Black. So two helpful ways to think about genetic diversity within ancestry groups. So within ancestry groups is, the first one's heterozygosity. So for those of you not as familiar with genetics, heterozygosity, it means that at a particular location, in the chromosome, one copy that you got from your mother and one copy that you got from your father are not identical to one another. Okay, so heterozygosity means you have two copies and they're not identical. So we could think about diversity within an ancestry group in terms of heterozygosity by saying the more sites where individuals are heterozygous within that population, the more genetically diverse their populations are. Okay, so let's just look at this image again and think about that. So if I'm in population A, or no, I'm sorry, if I'm in population 3, I am going to be homozygous at this location where A occurs, right? Because A is the only allele that occurs in that particular population. So the chances are nearly 100%, right, that I'm going to inherit an A from a biological mother and an A from a biological father, okay? So if you look at that across the entire genome, and I repeatedly have homozygous SNPs, I got the same copy from my mom and my dad, that means there's not a lot of diversity in the ancestral group that I'm from, okay? Whereas, you know, if I'm in population 1, I'm likely to inherit a copy of A from one parent, a copy of B from the other parent, and therefore that implies that the population at that location at least is split, right, between those two alleles. And if that occurs repeatedly throughout my genome, that means I'm from a relatively genetically diverse population, okay? I'm going to get there about why I think diversity is such a helpful concept, okay? The other way we can think about genetic diversity is by thinking about private alleles. So private alleles are SNP sites that are biallelic in one population or triallelic, but monoallelic in all other populations, okay? So that means, let's say, most populations throughout the world only have an A at a particular site. But in my population, we have both A and B. So that is another sign that the ancestral group that I'm in or that I'm from has a lot of diversity in it. Okay, so now let's talk about why that matters, okay? So if you look across populations that live in Africa in modern times, in today's world, there is way more heterozygosity than in any of the rest of the world. So the orange populations in this image are the populations that today live in Africa. And the height of the bar shows you the frequency of heterozygosity in each of those populations. Okay, so you can see that populations within Africa are have a lot of genetic diversity. They have a lot of locations where some people have A, some people have B, and that repeats over and over and over throughout the genomes of those populations, okay? And as you move to the right, you can see that the amount of heterozygosity drops off, okay? The reason that there's so many orange populations on this slide is that this is by my colleague, Sarah Tishkoff, who sent me this slide. She works with African populations that live in Africa. So this paper was really focused on Africa. Okay, so we look at private alleles. We can see that populations that today live in Africa have a great deal more sites, SNPs, where those populations have 2 alleles at that site, but every other population in the world only has 1 allele, okay? So, and you can see that this is quite striking. So the point that I'm driving home here is that ancestral groups from what is today Africa, are much, much more diverse than the entire rest of the population. And you can see here, this is also from Sarah Tishkoff, and but this was a version of her images that they used at the New York Times. And you can see the dark color, the dark green color on this slide is more diversity. The light green color is less diversity. And in this case, they're showing how this can be used to infer movements of ancient groups of humans. But the point that I want to make is ancestral groups that live within Africa are much more diverse than the rest of the population of the world, okay? And as a matter of fact, people like Sarah Tishkoff could tell us African ancestry groups reflect more genetic diversity than the rest of the global ancestral groups combined, okay? So this is super striking, and I'm now going to tell you why I think that is important. Race is a way of thinking about groups of people based on socio-political categories. But despite the very confusing language that we use sometimes, like African American, race does not correspond well with genetic ancestry. So, this slide tells us that people who today live in Africa are likely to be more genetically distinct from one another. 2 people living in Africa today are more genetically distinct from one another than one person who currently lives in Africa is from a person who is of Native American heritage. Okay, so there's more diversity within Africa than the entire rest of the population. 2 people who live in Africa today are of African ancestry are more dissimilar from one another than either one of them is from a Native American. Okay. So let's look at now American racial populations and ask the question, what can we know about the ancestry of people who today live in America? Okay. So this is a study of individuals who self-identify as Black in America. Okay. And you can see here, let's look at A. The color scale here is an estimate of the African ancestry as a percentage for these individuals. You can see, for example, in South Carolina, an individual who self-identifies as Black is likely to have more than 80% of their ancestry coming from the continent of Africa, okay? If you look at Kentucky, a person who self-identifies as Black in Kentucky has somewhere between 0.65 and 0.7, or 65 to 70% of their ancestry coming from Africa. So just for comparison, let's look at C. So a person living in West Virginia who identifies as Black may have more than 30% of their ancestry from Europe. A person who lives in Kentucky who self-identifies as Black may have somewhere in the range of 25 to 30% of their ancestry coming from Europe. And you can see B shows something similar, but for Native American ancestry. Okay, so the point I'm trying to make here is that the racial categories that we use in the US to discern between groups of people that we perceive to be different in socio-political terms, Ancestry does not correspond to that, okay? Just for comparison, this is self-identified white Americans, Americans who self-identify as white, okay? And you can see that this is a slightly different kind of image. It's showing you among self-reported whites or European Americans, what percentage of those people have greater than 2% African ancestry. And you can see in Louisiana and South Carolina, more than 5% of Americans who self-identify as white have greater than 2% African ancestry. And B shows you the same thing, but for Native American ancestry, okay? So the point I'm trying to make here is American residents, people who today live in America, reflect a significant amount of genetic admixture, which is to say their ancestry is not from one group, it tends to be from multiple continents. And then race and ancestry don't correlate conceptually, right? We've talked at great length about how ancestry is determined. That doesn't correlate with what race is, which is a socio-political category. But just as importantly for our clinical practice, race and ancestry don't correlate biologically, okay? So let's talk about this a little bit in terms of implications for pediatric clinical practice. Okay, so I just wanted to give you an example. Our colleague John Cohen gave me this example, and it's just one way to think about the way that we currently use race in clinical practice. So this is data showing the infant survival probability, okay, at 24, 26, and 28 weeks, A, B, and C, between Black females, white females, Black males, and white males. And for the purposes of this study, race was defined by self-report by the mother, okay? And you can see that when we separate out those four groups, the survival for Black females is highest, and the survival for white males is the lowest. And as a matter of fact, if you look at it from a number perspective, just among those infants who are less than 1,000 grams, Black females are more than twice as likely to survive than white males, okay? The reason I think that this is so important is the way that our clinical brains work is that we look at this and we unfortunately assume that there must be something biologically inherently different between Black females and white males. If their survival is so different, then there must be something going on there that is like in our genes, right? So this is just one example of how frequently we tend to use race in pattern recognition in clinical care, okay? But this is deeply problematic, and I'm just going to go through 8 reasons why I think that this is problematic. The first couple are just restating what we've already explored, but I just want to put them into this context, okay? Number one, self-reported race is not dependably associated with genetic ancestry, right? We just looked at that in the American context. The same is true throughout the world, actually. Okay, so self-reported race is not dependably associated with genetic ancestry. Perceived race is even worse. So when we have patients, I asked in our clinic, the front desk documents race in EPIC, but they tend not to ask the parent, what is your race? because they find that awkward to ask. So they tend to document in the chart what they perceive the parent or the child's race to be. And then we do this in clinic all the time. As a matter of fact, when we present cases, right, we say, this is a 13-year-old African-American male. And we are taught to do that every single time. But actually, the vast majority of the time, we didn't even ask the patient what their self-identified race is. We just give our perception. So as bad as self-reported race is in corresponding to genetic ancestry, perceived race is even worse. And what makes perceived race additionally unhelpful is that the self-reported race of an individual is a better measure of the effects of socio-political factors, systemic racism, social determinants of health on the patient's health than perceived race is. Perceived race is unrigorous, and we should stop using it. Okay, number 3. From a genetic perspective, individuals of African ancestry are potentially more dissimilar from one another than they are from members of other ancestral groups. Okay, so when we looked at the slide of infant survival probability between different American race and gender categories, the chart was made that way because we thought black females are similar to one another biologically. But from a genetic perspective, black female girls can potentially be more dissimilar from one another than they are from members of other ancestral groups. Okay, so our perceptions of race correspond very poorly with the genetic ancestry, the underlying genetics of the patients that we take care of. American residents reflect a significant amount of genetic admixture. Even though we associate races with a continent and we even use terms that imply that, in fact, we are, to a large part, descendants of more than one ancestral population, frequently from more than one continent. Number 6, this is moving into some new territory that we haven't discussed yet. Differential diagnoses driven by racial assumptions will exclude important diagnoses. So this is a case reported in Pediatrics in 2003. This is just a discussion on race, but this is an anecdote given by the author. My childhood friend Leela wasn't diagnosed with cystic fibrosis until she was 8 years old. This is obviously before newborn screening. She had all the symptoms, and it was repeatedly, no one suspected cystic fibrosis. And then here at the last sentence, only when she was 8 did a radiologist who had never seen her face to face notice her chest radiograph and ask, "Who's the kid with CF?" So there are actually multiple examples of diagnoses being failed to be made because the clinician taking care of a patient assumed that a condition was associated with one race group. But remember, race and genetic ancestry don't correspond well. So it is critically important for us not to miss diagnoses, for us to stop using race as part of our pattern recognition. This is somewhat overlapping with the last one, but it's saying in a slightly different way. Medical conditions, including genetic conditions, do not track with race in the ways we tend to assume. So just think about cystic fibrosis patients. Think about sickle cell patients, okay? What's the ancestry of sickle cell patients, right? So sickle cell variants that cause sickle cell disease are passed down ancestrally, right? They are passed down through generations. And we associate that variant with the continent of Africa, right? check this out. Some of the populations in the world with the highest allele frequency for the variant that causes sickle cell disease are in India, right? You can see here there are ancestral populations in Europe that have the sickle cell variant, in the Middle East, in Africa, on Madagascar, and in India. This is called the malaria belt, right? So we make assumptions that, oh, we should look for sickle cell in individuals who are black, who self-identify as black. But if we do that, we're guaranteed to miss the occurrence of this disease in individuals who don't meet our expectations, right? We have to separate race and pattern recognition. When we use race in clinical pattern recognition, we tend to reinforce our own incorrect assumptions about the biological basis of race. This is kind of the topic of this whole talk, okay? We assume that there's something biologically different between the patients that we take care of who self-identify as black and those who self-identify as white, but that is scientifically and biologically a misperception. Okay. But we know that health disparities across racial and ethnic groups are real and are of critical importance in our efforts to improve the health of all children. So how do we resolve that tension? We know race and ethnicity are important to health, but our assumptions about race don't correspond to biology in the way that we expect, right? So how do we resolve this tension? These are just some suggestions that I have, and this will be heading towards the end of this talk. So number one, stop asking our students to include race by default when presenting patients. The problem here is that it is a constant reminder to us that race is biological, right? We put it in with age and gender as like the key demographics that you have to know in order to know sort of the biological background for the patients we're taking care of. But that is a misperception, and I think it would be really helpful if we stopped focusing on race. It'll make us better at recognizing patterns. If teaching about the apparent association of a medical condition with a racial or ethnic group, ask yourself, how strong an association is this, and do I even understand the mechanism for this association? In a great number of cases, there is a disparity because of the kinds of assumptions that we tend to make. We assume that there's something biological there, about there's a difference in gene frequency, or there's a difference in a disease-causing gene that happens to be carried in a particular ancestral group. But typically we don't even understand, that's our assumption. We don't have any real evidence for that. And often the association is not as strong as we think it is, right? So just, if you're teaching about an apparent association of a medical condition with a racial ethnic group, think twice about whether you really understand what underlies that. Number three, assume the mechanism for a health disparity is due to social determinants of health, not genetics or other biological factors, until proven otherwise. You'll almost never be wrong. The vast majority of the health disparities that we encounter in the care of our patients have to do with chronic stress caused by social determinants of health, including systemic racism. So, which can have obviously profound effects on biology, right? But they're not genetic. The underlying dynamic, the underlying mechanism is a social determinant of health, or a combination of social determinants of health. Number four, when building a differential diagnosis, never exclude something because of the patient's race or ethnicity. Okay, discount any clinical decision support tool that incorporates race, since race is an extremely poor way to represent probabilities related to an individual patient, right? So if a social determinant of health, or for that matter, if a biological mechanism underlies a decision that we're making, we should be using that and not using race as a proxy for that. 6, don't document a race unless it is a category the patient reported themselves or the parent reported on their behalf. Okay. All the time we document our perceived race, and again, we're just reinforcing our own incorrect assumptions about race by doing that. Okay, just a few more thoughts, going a little bit beyond clinical care, but I wanted to say a few things about this because really, you know, what we do in our everyday life, it sets habits and kind of sets up the way that we think in ways that affect our clinical decision making. Okay, that's just a polite way of saying our pre-existing biases affect our clinical decision making, right? Okay, so avoid thinking in terms of genetic essentialism, right? When you see Kyle on the Ancestry.com commercial say that because of his genetic ancestry, he changed the celebration of his heritage, that's a genetic essentialism. It's saying that this is a quote from James Watson of Watson and Crick fame, we used to think our future was in the stars, now we know it is in our genes. Don't get caught up in that kind of thinking. I'm not saying genetics is irrelevant to our health, it clearly is, but genetics operate in health primarily on an individual and family basis, not on a racial basis. And fundamentally, our ancestry does not determine who we are. And then I know we've talked about this before in other grand rounds, but I just want to be really clear about it. Learn about systemic racism and consider how it might influence disparities in your specialty. And that will help you, I think, in the long run, start to break out of that way of thinking that disparities have some biological or fundamentally biological differences, whereas in fact, if we could alter the environment, the systems of racism that operate in our society, we could alleviate those disparities. And unfortunately, some of those mechanisms actually occur in our own interactions with patients and in our clinics. So we do have power over breaking down at least some of those factors. Okay, and then have fun with genetic ancestry tests if you like. I've never gotten mine, but maybe I will someday. They are pretty good as far as they go, but remember that a human being is far more than their genes. So I'll just leave you by talking about the bad Kyle here, the other Kyle. Merker, M-E-R-K-E-R, is a German name, and as you know, last names tend to be passed down in a patrilineal way. So old Kyle probably celebrated his German heritage because one of his paternal ancestors was an adoptive father, or perhaps, you know, the person he thought was his father was actually not his ancestral father, his biological birth father. So just remember, you know, if that paternal ancestor of Kyle Merker's was German and passed on that German heritage, why should Kyle think that's not part of his heritage, right? We're way more than our genes, and our racial identity, our the heritage that we celebrate doesn't have to be, and is often not, tied to our ancestry. Okay, so I'll stop talking there. I think we have time for a few questions. All right, we'll stop sharing. Okay, I see Jeff's comment. Okay. So Dr. Roberts asked, how is the diversity within the African continent explained? I would have thought that migration and travel over the millennia would have created more diversity elsewhere in the world. Also, the graph of sickle cell populations suggests that the sickle cell mutation did not arise in one individual and because of evolutionary advantage spread by selective advantage, but rather the mutation occurred in various populations de novo. I'll answer your second thing first. I agree with you, Dr. Roberts, that the graph of sickle cell population suggests that the variants that cause sickle cell did not arise in a single individual. They emerged actually multiple times in multiple populations. And that is supported by the evidence that shows that the actual genetic mechanism that leads to sickle cell in those populations is not identical. It's actually different, okay? So the first one, how is the diversity within the African continent explained, is this is the grandest example of the founder effect in all of human history, right? So African populations are, for all of us, the ancestral population. Again, a little bit of pointing out how silly it is to say my ancestry is from Scotland. Actually, my ancestry ultimately is from Africa, right? So the African, all of the population of Africa did not migrate out of Africa and people the rest of the world. A large proportion of the ancestral groups that live in Africa, stayed there. They didn't spread their genes outside of Africa. So that's why Africa has so much genetic diversity. It's also the evidence why we think our ancestral populations are from Africa. Thanks very much, Dr. Allele. Okay, and thanks, Sarah. I appreciate that. Any other questions, folks? Nicole's asking, I agree so strongly with this presentation and your message. Do you think it is worth acknowledging race for the purpose of recognizing it as a potential source of difficulty by experiencing racism as an adverse childhood experience? Exactly how we define ACE is not within my area of specialty, but I do think it is important for us to elicit race, which is to say, ask about self-identified race of our patients. It is important for us to at least think about systemic racism as the mechanisms for causing conditions. So yes, I do think that. Whether we make it explicitly part of ACE's measurement, I'm not an expert on that. So John Birkenbach says, still seems to me that these ancestral genetic tests work from backwards logic. They take the genetic makeup of a population living in an area right now when that area has had centuries of population migration beforehand and subsequent inter-quote racial propagation, etc. John, that's exactly right. And taking that even further, it's basically, these tests are all based on where populations live today, not on where they lived when my ancestors were a part of that population, right? So just one clear example is Anglo-Saxons, right? Those are the peoples, one of the peoples that lives in UK. Saxony is in Germany. The peoples of it who identify as Anglo-Saxon heritage actually have ancestors that were early residents of the British Isles and also a later introgression of German populations. So anyway, these are clearly based on backwards logic. You're exactly right. And in many ways, they are taking what we today recognize as sort of political boundaries and putting them on top of something that those boundaries haven't always been that way. Okay, I think we're low on time. If anybody, I'll talk about this till the cows come home. So if you ever want to discuss this, stop by, send me an email, whatever. I'd be more than happy to discuss it. I love geeking out about this. Okay, thanks so much, everybody.
Thank you. Nice presentation.
Thanks so much. All right, Kai.